त्याच्या व्यतिरिक्त काही सॉफ्टवेअर्स असतात जे कंपनी टिपिकली यूज करतात, ते तिथे लिहिलेले आहेत. ठीक आहे. काही प्लॅटफॉर्म्स असतात, ठीक आहे, ते तिथे लिहिलेले आहेत. लॅब टेक्निक्स तुम्हाला माहिती आहे, एलिझा, वेस्टर्न लॉ वगैरे, राईट. डेटाबेसेस असतात, हे जे डेटाबेसेस लिहिलेले आहेत हे तुम्हाला माहिती नसते, कारण हे ना प्रॅक्टिकली यूज केल्या जातात, पण ते आपल्या सिलेबसमध्ये नसतात. कारण सिलेबस असतात ते 5-6 वर्षांनी लिहिलेले असतात किंवा त्याच्या आधी लिहिलेले असतात. ठीक आहे. तर मग जेव्हा असे जॉब येतात तर ह्या सगळ्या गोष्टी तुम्हाला गुगल करावं लागतात. ठीक आहे. या सगळ्या गोष्टींबद्दल तुम्हाला माहिती काढा. प्रीक्लिनिकल ड्रग डिस्कवरी रेट.
Preclinical research associated key focus support animal studies, toxicity, profiling and pharmacokinetic evaluations, toxicology assistant, help access compound safety, dose response and organ specific toxicity, pharmacology lab technician, conduct in vivo and in vitro efficacy studies and behavioral assay, bioanalytical training, assist in sample preparation and LC-MS analysis for drug concentration data, formulation, R&D intern, preclinical, development prototype of animal doing and stability testing, pathology support assistant, help with histopathology slide, pre and organ tissue analysis, preclinical documentation coordinator, maintain study protocols, lab notebooks and regulatory records.
याच्याबद्दल कोणाला काही प्रश्न आहे? ही तुमची अपॉर्च्युनिटी आहे, जे तुम्हाला समजलं नाही ना ते विचारू. कारण ह्या जे आहे, ठीक आहे, हे तुम्हाला सगळ्याच जॉबमध्ये जे आहे, ठीक आहे, हे तुम्हाला सगळ्याच जॉबमध्ये आता यूज करावं लागतं. AI किती लोक वापरतात? इन्स्ट्रुमेंटेशन स्किल्स, आता ते प्रत्येक इन्स्ट्रुमेंट्स वेगळे वेगळे असतात. त्याचे जे थेअरी असते ती तुम्हाला शिकवण्यात येते, प्रॅक्टिकल तुम्हाला मिळणार नाही. काही काही प्रॅक्टिकल आहे, पण जे तुमच्या लॅबमध्ये आहेत ते फार्मा कंपनी सहसा यूज नाही करत, ते आउटडेटेड झालेले असतात. पण त्याचं तुमचं बेसिक नॉलेज किती आहे ते तुमचं टेस्ट केलं आहे. आणि लास्टली बेसिक मॉलिक्युलर बायोलॉजी. आता टूल्स ज्या वापरल्या जातात, HPLC, LC, UV, आजच सर मला त्याच्याबद्दल बोलत होते. UV बद्दल तुम्ही आजच बोलत होते ना मला. डॉक्युमेंटेशन सिस्टीम, लिम्स, LEM. लिम्स बद्दल किती लोकांना माहिती आहे? पण जर का प्री क्लिनिकल रिलेटेड जॉब आला, ठीक आहे, तुम्हाला ह्या गोष्टी गुगल करावं लागते. त्याच्याबद्दल एक छोटंसं इंट्रोडक्शन तरी हे करावं लागेल. कारण जेव्हा तुम्हाला कोणी विचारेल की हे काय आहे, तर तुम्ही असं नाही म्हणू शकता तुम्हाला माहिती नाही माझ्या सिलेबस. तुम्हाला थोडंफार तरी त्याच्याबद्दल काहीतरी माहिती द्यावी लागते. GLP तुम्हाला माहिती आहे. OECD गाईडलाईन्स किती लोकांना माहिती आहे? गुगल करावं लागेल. ठीक आहे, माहिती करावं लागेल. त्याचे क्रायटेरिया जे आहेत ते सेट करून ठेवावं लागेल. ओके. सॉफ्टवेअरमध्ये एक्सेल सगळ्यांना माहिती असेल. ग्राफ पॅड रिझन नाही माहिती, ते बऱ्याचशा इंडस्ट्रीज यूज करतात. आणि केम ड्रॉ पण माहिती आहे. आता एक रोल आहे. आता काय काय डिस्कवरीमध्ये काय जे रोल्स आहेत ते खूप डिस्कवरीमध्ये जनरली कसं होतं म्हणजे की फार्मसी जेव्हा कोणी बीफॉर्मला फार्मास्युटिकल कंपनी हे करतात, तर मॅक्झिमम लोक फॉर्म्युलेशनमध्ये घेतात. ठीक आहे, फॉर्म्युलेशन किंवा प्रोडक्शनसाठी जातात. बरेचसे लोक जे आहेत ते क्लिनिकल रिसर्च अँड डेव्हलपमेंटमध्ये मोस्टली जे ह्युमनमध्ये क्लिनिकल ट्रायल्स होतात त्याच्यामध्ये जास्त ॲब्सॉर्ब होतात. प्री क्लिनिकल आणि डिस्कवरी साईडला कमी होतात. तर त्यामुळे मी काही काही जॉब प्रोफाईल्स इथे असे क्रिएट केलेले आहेत की जे रिसर्च असोसिएटेड प्री क्लिनिकल R&D आहे ते काय करतात.
Research associated R&D role support formulation, synthesis and analytical development. Task assist in designing experiment, preparing sample and documenting lab result. Skill needed: basic lab techniques, GLP knowledge, attention to detail. Primary role a research associated in preclinical R&D support laboratory-based studies that evaluate the safety, pharmacokinetics, and pharmacodynamic of new drug candidates before they enter human trials. This role is hand-on, technical, and foundational to the drug development pipeline.
काय प्रश्न क्वेश्चन
A research associate with clinical R&D
same job for responsibility
for responsibility, experimental design and execution, assist in planning and conducting in vitro and in vivo experiments, prepare formulations and dose solutions for animal studies, operate lab industries like instruments like SPLC, GCUP, NMR and GPC for sample analysis. Formulation and synthesis support. Develop and optimize drug formulation for preclinical testing. Synthesize and purify chemical compounds or polymer conjugates. Characterize formulation for stability, solubility and bioavailability.
right yes
Research Associate, Core Responsibility, Toxicology, Biocompatibility Studies, Support GLP compliant, Toxicity Evaluations on Animal Models, Document Observation, Dose Responses and Adverse Effect Contribution to Risk Assessment and Regulatory Documentation, example CBS and ECT, Data Analysis and Documentation, maintain meticulous lab records and raw data logs, analyze results using statistical tools or in silico models, prepare technical reports, summarize and SOPs for internal review, regulatory and quality compliance, ensure adherence to GLP, OECD, ICH and ISO standards, participate in audits and inspections, support documentations for regulatory submissions and safety evaluation.
question is what meticulous lab records. What is it? Now, lab records, all of you know. Right? If I am conducting an experiment, then I have to write the date. Then I have to write what I am doing. That one has to be kept till the drug gets approved. Because when the auditor comes, they will try to know that how much water I have put, how much chemical I have put, what I have done. All that thing is written there. So when they are tracking and when they are doing audit, they will come and then try to find out whether I have really done it or not. Now on top of that, most of the pharmaceutical labs have biometrics entry and exit into the labs. So while going, you swap it, you go there, you do your experiment, then you come back and then do it. Now the auditor would say, the date is written here this one. HPLC was run like this. HPLC has 100 ml water. While notes it is written only 10. Then there is a discrepancy. So when such thing has to be taken care very minutely, because each detail there is a chance that is going to get rectified by auditor, and the auditor is going to question everything what you do. That's why the lab record has to be meticulous, because it is going to get cross-checked. Because the drug what you are developing is going to go to some human being, and they may have a toxicity, right? So that's why the regulators require all these records has to be done. This looks very simple. Whatever I am doing, I am just writing it down. It is not that simple job because they have not put their lab records properly. When I was working with Cipla, we used to really request the pharmacies to keep their lab records up to date. आज कर लिया, कल कर दूंगा, कल लिख दूंगा, ठीक है, लिखना ही तो है, right? These things are very common in industry, and this becomes a major audit findings, and sometimes the approval of the drug gets rejected because the people who were working on it does not get their diaries properly. Okay, so this is one of the, when you start working in a industry, one of the things which will be expected from you is whatever you do, you have to document it. You have to find good documentation practice. If there is something you do something wrong, you strike it, then write it, then put your initials in there, right? Your records would be there till drug gets approved. It is going to get scanned. It will be always there. Okay, so it is very important. 90% of your work, at least in first 2 years, will be related to lab, and all you have to do is whatever you are doing, you have to write it down. Okay?
Toxicology assistant role: help conduct toxicity studies on animal models. Task: dose administration, observation, data recording, and sample collection. Skills needed: understanding of pharmacology, animal handling, safety protocols. Primary role: a toxicology assistant supports laboratory operations in evaluating the safety and biological effects of chemical substances, drugs, and formulations. Primarily in preclinical settings, this role is crucial for identifying potential toxic effects before human trials begin.
Any questions? Answer me.
Core responsibilities: sample preparation and handling, prepare biological and chemical samples for testing, example blood, urine, tissue, label, organize and store samples according to SOPs, ensure proper disposal of hazardous waste and biohazards, conducting toxicological tests as well as assisting acute, subchronic and chronic toxicity studies, monitor animal models for behavioral and physiological changes, record observations related to the dose response, organ toxicity and mortality. Reagents and equipments management, prepare reagent and solution for assay, calibrate and maintain lab instrument, example spectrophotometers, centrifugal tubes, sterile and organized lab equipments to maintain cleanliness and accuracy, data documentation and reporting, record test results and observe in lab notebooks or digital system, assist in compiling data for toxicology reports and regulatory submissions, follow good laboratory practices, that is GLP, and ensure audit readiness, safety and compliance. There are 2 safety protocols and headline hazardous substance, wear appropriate PPE and follow biosafety guidelines, support inspections and maintain documentation for.
What is PP? Something related to personal equipment, right? That is also a very big important thing. See, when we think about these things, we always think these are very simple things. Okay? But in big companies, where does an employee washes his hand? You must have seen in restaurants that employees has to wash a hand with this toilets, everything, right? In pharma world, it becomes 10 times, 20 times. That all employees, if there is a clinical reaction happens, and if the fumes are going into his eyes, what he has to do? First thing is that he has to wear the glasses, right? He cannot enter there without glasses. For some reason, he is a hero and then he keeps his glasses here and start working. Okay, fumes goes to him. Then where he has to run, right? And that water system has to be available so that in one place the water should go in on his eyes. So all those things are very very important. We take it for granted. ये तो क्या, ये तो मेरे को मालूम है, ये तो मैं कर लूँगा. नहीं, जब होता है ना तो किसको मालूम नहीं रहता. ठीक है? So that is very important. And these are the very small things, but this sets you apart. Okay? When someone is asking you questions about, let's say, toxicological assessments. Okay? Now, these skill sets and everything, but when I am emphasizing on it, with all these points are going to get tested. Or you have to make sure that you tell all these points to them that they are know.
Analytical development executes role development and validation analytical methods for drug surveillance. Tabs use instruments like HPLC, GC, UV, assess purity of resolution and stability. Skill needed, instrumentation skills, methods, developments and documentations. Primary role, and analytical development executes responsible for the developing, validation and optimizing analytical methods. Uses to test pharmaceutical products. Their work ensure that drugs meet quality, safety and efficacy standards before clinical trials or market release.
Any questions?
Analytical development and execute. Core responsibility, method of development, design and define analytical method of raw material, intermediate and finished products, select the appropriate technique, example HPLC, GC, UV, FTR, based on the compound property, troubleshoot and optimize method for accuracy and reproducibility, method validation, analytic method as per ICH guidelines, specificity, precision, accuracy, linearity, robustness, document validation, protocols and report of regular compliance ensure methods are suitable for the routine quality control and stability study, stability and complexity studies. Conduct stability testing under the various conditions, temperature, humidity, light, analyze degradation, product and shelf life parameters, support formulation terms and conformity data.
Any questions? Next. Robustness. Robustness means very strong. कोई एक strong होता है, उससे भी जो ताकतवर होता है, perfect होता है, उसको robust. So everything has to be specific. Looking at specificity, precision, accuracy. Done properly, or whether everything is following a proper order. And the robust means that whole system has to be very strong and very perfect.
Core responsibilities, instrument calibration and maintenance. Calibrate lab instrument regularly and maintain the performance. Ensure instrument and audit ready and compliant with SOPs, troubleshoots, equipment issues and coordinate repairs, documentation and regulatory supports, prepare study protocols, validation report and technical summaries, achieve documents for SOPs and regulatory timelines, support regulatory submission, example, seat reading modules for IND and NDA fillings.
What is SOP? What is the first SOP written? Someone has to start writing SOP, right? So what is the first SOP? First SOP is always SOP of SOP. How the SOP will be written, who will validate it, how it is going to get executed, and who will follow it. So that is the first SOP of SOP. Then the system wise SOP starts. Okay?
formulation scientist, entry level. Role: assist in developing dosage forms for preclinical testing. Task: prepare tablets, capsules, suspensions, optimize excipients. Pharmaceutics knowledge, accounting skills, regulatory awareness. Primary role: an entry-level formulation scientist supports the design, development and optimization of pharmaceutical dosage forms. Their work ensures that products are stable, effective and manufacturable.
Board's responsibility, formulation studies, analyze physiochemical properties of active pharmaceutical ingredients, assess stability, pH sensitivity, hygroscopic compatibility with excipient, conduct literature review to guide formulation strategy. Dose form development, assist in developing tablet, capsule, suspension, emulsion, and injectable. Selected the test excipient for stability, biopredictive.
Responsibility, process optimization and scales. Responsibility, process optimization and scales. Collaboration with manufacturing team to a transition lab scale formulation to pilot batches. Troubleshoot formulation issue during scale up, support documentation and compliances. Maintain detailed record of formulation experiment. Documentation and compliances. Maintain detailed record of formulation experiment and JMP, GLP and regulatory